Hello everyone, this is Dr. Kellen, and welcome to another episode of Inflammatory Content, the podcast about immunology and biomedical research more broadly. I'm here to interview world-class researchers, to communicate broadly the cutting-edge work going on in their laboratories, and to get to know the people behind the science. But today, we're going back to our roots and doing a Journal Club episode. I have a really exciting immunology and genetics paper to share with you all today. The paper is a little bit outside of my area of expertise, so please write in if you think I got anything wrong. So sit back, relax, and let's dive into some science. And remember, if you enjoy the show, please consider sharing the episode, leaving a review, or donating to support our work. Thanks for listening. The paper we'll be covering today is called "A Disease-Associated Gene Desert Directs Macrophage Inflammation Through ETS2." This article was published in Nature in June of 2024. There are a number of authors on this paper from the Francis Crick Institute in London, in the UK, primarily. The first author is C.T. Stankey, and the last author is J.C. Lee. So why did I choose this paper? The authors used genetics in a very innovative way to understand how the immune system works, making it a landmark paper. Moreover, I'm really interested in this paper because I'm interested in using some of these approaches that the authors use in my own work, and the best way to learn something is to teach it, so here we go. Now, before we get into the paper, I'd like to give some background on genome-wide association studies, which will come up in the paper. These are abbreviated as GWAS, and since the first one was published in 2005, these studies have identified many SNPs, SNPs, which is short for single nucleotide polymorphisms or variants that are associated with various diseases, immune diseases or otherwise. However, most of these variants lie in non-coding regions of the genome, regions that don't give rise to protein. So it's unclear which genes these variants regulate in which cell types and what they do. Overall, this has led to a poor translation of GWAS findings into therapeutics. There are many challenges of interpreting genome-wide association studies. For one, there is often high linkage disequilibrium between variants. This means that the variants are very close together on the chromosome, leading them to not recombine during meiosis. This leads to individuals having many of these risk variants together, and we call this the risk haplotype. It's the collection of risk variants. So you don't really know which variant of all these variants in this region of interest is actually causing the problem. Additionally, there's variable levels of regulatory activity of the genomic regions across cell types. So which cell type is actually causing the problem? And third, there can be multiple genes within the associated locus, making it difficult to understand what the causal gene is. One potential solution to this problem is the field of functional genomics. In terms of identifying the causal variant, statistical methods can be used, such as fine mapping, to identify the causal variant from all these potential variants. Additionally, functional approaches like CRISPR-Cas for genome editing and reporter assays can be used to figure out the function of these variants, actual experiments in the lab. In terms of identifying causal genes, promoter-enhancer interaction experiments can be performed, like Hi-C, that looks at organization of the genome to see which gene that variant is affecting. And then in terms of identifying the causal cell type, you can do regulatory element analysis using approaches like ATAC-seq, which is assay for transposase accessible chromatin using sequencing, and ChIP-seq, chromatin immunoprecipitation sequencing, to look at whether specific cell types possess regulatory elements within the risk haplotype. And also to identify causal cell type, you could use EQTL data, which is expression quantitative trait loci, and use this to determine whether the risk haplotype is associated with changes in gene expression in specific cell types, helping to identify the relevant cell types and mechanisms. So the big question these authors aim to address in this paper is, can functional genomics be used to better understand autoimmune and inflammatory disease and identify promising therapeutic targets? The paper mostly focuses on inflammatory bowel disease, but the approach could be used for any sort of condition. The authors first looked at publicly available genome-wide association study data. These data are from 4 different inflammatory diseases: inflammatory bowel disease, primary sclerosing cholangitis, ankylosing spondylitis, and Takayasu's arteritis. And they found in this specific region of chromosome 21q22, so that's the region of chromosome 21, that there are SNPs that are associated with all 4 of these diseases in a non-coding region. And if you line up the Manhattan plots, you can see that all of these variants across these 4 diseases line up, and they find that there's a significant co-localization across these diseases, suggesting that there's a shared genetic basis. Now, they next leverage more publicly available data to ask the question, does this region contain a distal enhancer that works in immune cells? And so here they're looking at publicly available H3K27 acetylation ChIP-seq. This is histone 3 lysine 27 acetylation. This marker is indicative of active promoters or enhancers, and these are regulatory elements that are important for transcribing DNA into RNA. And by looking at these data across different cell types, monocytes, B cells, T cells, neutrophils, they see that only in monocytes are there active enhancers at this non-coding region. So really seems to be monocyte specific. Now they next ask what genes are regulated by this enhancer. And so here they use macrophage, more publicly available data, by the way, macrophage high C data, which is the 3D structure of the genome. So we can actually see which parts of the DNA are interacting with each other. There are notably 3 genes next to this risk haplotype, ETS2, BRWD1, and PSMG1. ETS2 being the most distal of these 3 genes. And with these high C data, they see that there's a physical interaction with this gene ETS2, which is actually a known transcription factor, meaning it binds to DNA and regulates transcription, and it has a controversial role in macrophage biology. Some studies show that it promotes the inflammatory response in macrophages. Some show that it does the opposite. So it's a little fuzzy about what this gene actually does. And then with the EQTL data, they show that if you have the risk haplotype, you're going to have higher ETS2 expression, further suggesting that ETS2 is a causal gene. So the next question the authors ask is the enhancer, which is, remember, where the variant is, required for macrophage expression of ETS2? And to address this, they actually do an experiment now. Everything else before this was leveraging publicly available data. And here they're doing a CRISPR deletion of the enhancer in primary human monocytes and then differentiate them into inflammatory macrophages. And they do this by giving the cells GM-CSF plus a cocktail of TNF, prostaglandin E2, PAM3, CSK4. They argue that this is the most relevant way to activate a macrophage. Before they actually look at the effect of deleting the enhancer, they want to show how they can look at the expression of these nearby genes. And they do this by an RNA flow assay. This is a way to look at gene expression by flow cytometry. I've actually tried this once and it didn't work too well for me, but it looks like it works pretty well for these guys. You see ETS2 go up nicely, BRWD1, PSMG1, these nearby genes, their expression goes up as the macrophages differentiate into their inflammatory state in unedited cells. But when you knock out the enhancer, where the variant is, only ETS2 expression goes down. BRWD1 and PSMG1 stay the same. So this further demonstrates that ETS2 is the gene that is being acted on by this enhancer, or at least one of the genes. Now, the next question the authors ask is, what is the causal variant in that enhancer? And so first they try to do one of these statistical fine mapping approaches, which did not work, I guess because the SNPs are too close together, but their analysis basically said there's an equal chance it could be any one of the 7 SNPs in that region. So they next do a functional experiment, and this is a massively parallel reporter assay, or an MPRA assay. And here they are generating a library of very short DNA sequences that are coupled with a minimal promoter, and it's a minimal promoter because it needs an enhancer to activate transcription, plus a reporter gene, plus a barcode. So they're going to transfect this library, which is going to tile along the entire region of interest of the genome, so they can test every little region, and they're going to transfect that into primary human macrophages, some with the risk allele and some with the non-risk allele. And then they're going to measure the expression of the reporter gene for each DNA sequence with and without the risk allele. The idea being that once you hit the enhancer, you're going to get high expression of the reporter gene. So here they tile along the entire length of that chromosome 21 region of interest. They're going across it, and they hit a couple different variants, and the expression doesn't go up that much of the reporter. Then all of a sudden, they hit this region where 3 of the variants are, and the expression shoots through the roof. And so they deemed that this region is the enhancer. So they now have narrowed it down from 7 potential variants to 3 variants. And then to put the nail in the coffin and find the causal variant, they look at the expression of the reporter gene in the risk versus non-risk allele and compare it for these 3 SNPs. And they see that only in the SNP RS2836882, only in this one, do you see a stratification between risk and non-risk. So this variant is the causal variant. Now, they next ask whether the SNP affects PU1 binding. PU1 is a pioneer factor in macrophages. It is a lineage-determining transcription factor. This means it is very critical for macrophage function, macrophage identity. And pioneer factor, think of it as a pioneer. It's going to go in before anyone else. It's actually going to bind to dense chromatin. Normally, transcription factors need accessible chromatin, loose chromatin to bind, but a pioneer factor can bind to dense chromatin and create open chromatin by binding, allowing other transcription factors to come in. And so here they're looking at ChIP-seq data, looking at all the binding sites of PU1. These are from heterozygous macrophages, again, leveraging publicly available data. And here they can see all the ChIP-seq peaks in this area, and they do a motif analysis looking for the PU1 motif. So where is that PU1 going to bind specifically? And they see that the SNP lies close, but it's actually outside of this canonical PU1 motif. So maybe it could affect PU1 binding. In the PU1 ChIP-seq, there is more binding of PU1 when the risk alleles. They're actually 4 times more. It's correlational, but it supports that this SNP promotes PU1 binding. And then they look at more publicly available data from heterozygous macrophages. This is now ATAC-seq, so this is chromatin accessibility. How loose is the chromatin? And they see increased chromatin accessibility with the risk allele. So you can actually stratify this based on the strand, and you can see that the strand with the risk allele actually has greater accessibility, which is consistent with increased PU1 binding. So they next do an experiment to confirm that the increased enhancer activity is specific to the risk allele, and they do this by doing H3K27 ChIP on risk and non-risk homozygous inflammatory macrophages. So now we're looking at 2 sets of macrophages that are homozygous for the non-risk and risk. And again, they see, confirming the prior analysis of publicly available data, that when you have the risk allele, you have greater H3K27 ChIP, so more active enhancer, confirming that the enhancer activity is specific to the risk allele. So to summarize that really dense amount of genetics in figure 1 of this paper, they found that this variant at chromosome 21q22 is an enhancer, and it's associated with several autoimmune diseases. It promotes binding of the pioneer factor PU1, increases chromatin accessibility, and increases or allows a physical interaction with distal ETS2 gene, this transcription factor, to increase ETS2's activity. Now, the rest of the paper is more immunology, less genetics. So they next ask the question, what is the role of ETS2 in human macrophages? And to address this, they do a CRISPR knockout of ETS2 in primary monocytes. And they find, using an ELISA approach here, so looking at protein levels, that the ETS2 knockout macrophages have significantly reduced pro-inflammatory cytokine production, like IL-1 beta, IL-6, IL-8. Anti-inflammatory cytokines like IL-10 were mostly not affected. In line with this decreased inflammatory response in the macrophages, they did a flow-based zymazan phagocytosis assay and observed significantly reduced phagocytosis in the ETS2 knockout macrophages, significantly reduced reactive oxygen species in a plate-based assay, and significantly reduced expression of genes related to reactive oxygen species. So overall, ETS2 is necessary for macrophage pro-inflammatory host defense immune function. And notably, there is no effect on survival or differentiation. To further understand the role of ETS2 in human macrophages, they did RNA sequencing to identify genes up and down in ETS2 knockouts. A bunch went down, but not too many went up. These genes that went down related to cytokine production, phagocytosis, Roth, just like they showed in the functional experiments. And by doing RNA sequencing on CRISPR knockout of the enhancer and correlation analysis, they found that the same thing happens whether you knock out the enhancer or ETS2. So knocking something out is a great way to figure out what it's doing, but another way is by increasing its expression. And so here they ask, what happens when you increase ETS2? And they did this by transfecting in vitro transcribed ETS2 mRNA into these primary monocytes. And I'd never heard of this as an overexpression approach, so I thought this was very clever. So they actually made ETS2 mRNA in a test tube and then transfected that into the macrophages. They also hit the macrophages with a low-dose LPS to get them a little angry first, and then looked at the response with and without the overexpression. And they validated that the ETS2 overexpression was actually working by gene expression. And then they did transcriptomics and found the same exact genes that we saw, or gene sets that we saw go down with ETS2 knockout, now went up with ETS2 overexpression. So it's not only necessary, but sufficient for the inflammatory macrophage phenotype, or sufficient in the context of that little LPS hit. They didn't show the data without LPS stimulation. So it's for sure enhancing the inflammatory macrophage phenotype. And the next asks, does ETS2 contribute to disease macrophage phenotypes? And so here they perform a gene set enrichment analysis with Crohn's disease inflammatory macrophage gene sets. Crohn's disease is a type of inflammatory bowel disease. And they see that a lot of the genes that are enriched in Crohn's disease inflammatory macrophages are highly expressed in ETS2 overexpressed macrophages, suggesting that the overexpression of ETS2 is relevant to the phenotype of macrophages in vivo. Now, to further understand how ETS2 contributes to macrophage phenotypes and disease, they compare this ETS2 overexpression signature in myeloid cells to other diseases, and they see that ETS2 is involved in multiple infectious and inflammatory diseases, particularly the ones that are associated with this risk variant that we've been talking about: Crohn's disease, Takayasu's arteritis, ankylosing spondylitis, and ulcerative colitis. They do not see so much of this signature in macrophages associated with tumors, but in infectious contexts like tuberculosis and sepsis, they do. So it's not a generic macrophage phenotype, but mostly associated with infectious inflammatory contexts. And that was all using publicly available data again. And the next thing they did to further understand the relevance to disease was a SNP-C analysis, which I had never seen before. So this is a SNP set enrichment analysis. What this analysis showed was that genes near inflammatory bowel disease associated SNPs are highly enriched in the ETS2 pathway. So the question still remains, how does ETS2 control macrophage function? And so here they did a co-expression analysis by using more publicly available data, a huge data set, 314N macrophages and monocytes from 67 different activation conditions. And they pooled all these together and looked, what is expressed along with ETS2. And they found at the very top of the list 2 genes, PFK-FB3 and HIF1 alpha, which are both very important for glycolysis. So maybe ETS2 regulates macrophages through metabolism. And to test this, they do a C13 glucose labeled metabolic flux assay in their ETS2 knockout and control cells and find that broadly glucose metabolism, glycolysis, the pentose phosphate pathway, the TCA cycle are all suppressed in the ETS2 knockout. They then test a HIF1 alpha stabilizer, which promotes glycolysis, and this does not affect the inflammatory phenotype. Yes, it switches these cells from OXPHOS to glycolysis, but it does not affect the inflammatory phenotype. So the question still remains, how does ETS2 control macrophage function? Now, to get at this, they take a epigenomics approach, and they look at what genes are targeted by ETS2. They use cut and run, which is a variant of ChIP-seq. They use it because the antibody that they have for ETS2 does not work in the context of fixation. So cut and run is a variant of ChIP-seq that will allow you to look at transcription factor binding with an antibody that may not work quite as well for ChIP-seq. And they find that ETS2 binds mostly to promoters, and these are marked by H3K4 trimethylation and also enhancers, the H3K27 acetylation. Not so much intergenic regions or gene bodies. So it binds to regulatory elements, and they validate this. They find canonical ETS2 motifs in these regulatory elements with the ChIP-seq data. So you do the ChIP-seq, you see where the transcription factor is binding, and then you're going to get all of these DNA sequences, and then you can run your motif analysis based on databases of these factors and see, okay, where exactly is that thing going to bind? So how does ETS2 control macrophage function? What genes are targeted by ETS2? Which promoters and enhancers is this thing binding to? And they find that it binds to gene promoters involved in the inflammatory response. For example, NLRP3 and TLR4, NOD2, a lot of pattern recognition receptors that are going to bind to PAMPs and DAMPs, pathogen-associated molecular patterns and damage-associated molecular patterns. And about half of the genes regulated by ETS2 have an ETS2 binding site in promoter or a cis-regulatory element. So a cis-regulatory element is a regulatory element like an enhancer that is on the same chromosome, but it could be very distal. So that suggests that ETS2 is directly regulating a lot of these genes, about half of them. They also note that ETS2 binds to the enhancer with the causal variant. Remember, they showed that PU1 binds there. So perhaps there's synergy that's occurring with PU1 and ETS2. Are they collaborating together? They next ask the question, is ETS2 and the genes it regulates expressed in vivo? And here they look at more publicly available single-cell RNA sequencing data from inflammatory bowel disease, specifically Crohn's disease. And they look at the myeloid clusters in these data, and they find that there are 2 clusters of interest here, the inflammatory macrophages and inflammatory monocytes, and both of these express relatively high levels of ETS2, and they are both enriched in the Crohn's disease patients over the healthy control patients. And not only do they just see high ETS2, but they have these gene scores that they've generated from their in vitro data of the genes that are regulated by ETS2. So they have this ETS2 module, and they can see that the ETS2 module is also high in these cells that express ETS2, these inflammatory macrophages and monocytes. They next look at another disease. This is now the primary sclerosing cholangitis, another one of the diseases that is associated with this variant. This is a liver gallbladder disease, and they do spatial transcriptomics here. They use the CosMx approach, which I personally have used as well. And I don't get a whole lot out of this experiment except to see that they found that macrophages near cholangiocytes highly express the ETS2 related genes. I didn't see any expression of ETS2 itself, but it looks like there's some inflammatory macrophages there, and they're trying to suggest that they may have high ETS2 expression, and that may lead to their inflammatory state. Now, they next ask, how can we therapeutically target macrophage ETS2? There's no known inhibitors of this transcription factor. And they did something very clever. They looked at this NIH links database, which I had never heard of. It is a database of 30,000 differentially expressed gene lists from cell lines treated with over 6,000 chemicals. So really a wealth of data here. And you can actually just plug in your module, your differentially expressed gene list, and see which type of chemical may regulate those genes. And they find that there are a bunch of hits that could potentially regulate ETS2. Some of them are known inhibitors or known treatments for IBD, such as JAK inhibitors and glucocorticoids. But there are also at the very top of the list a bunch of inhibitors that may regulate ETS2 that are not approved for IBD. And the one they really focus on is one of the top of the list is the MEK inhibitor. But there's also HSP90 inhibitors, RAF inhibitors, SARC inhibitors, ERK inhibitors. And they show that not only does just one MEK inhibitor, but a bunch of different MEK inhibitors all have this effect on the ETS2 module of downregulation. And now they test the effect of the MEK inhibitor in vitro, and they see that it phenocopies the enhancer in ETS2 deletion and downregulates inflammatory pathways in primary human macrophages. Now, finally, to get to the most translational experiment of the paper, they look at the effect of the MEK inhibitor on tissue explants from IBD patients. And so now they're taking biopsies of inflammatory bowel disease tissue, intestine, and putting it in a dish and testing how does this MEK inhibitor hold up compared to the standard of care infliximab, which is a TNF inhibitor. And they look at cytokine secretion from these explants, and they see that the MEK inhibitor significantly decreases the concentration of many pro-inflammatory cytokines that are relevant to disease, TNF, IL-17, IL-6, etc. And it does so to the same effect as infliximab. So it's just as good as infliximab in this assay. And they also do more analysis at a transcriptomic level here using bulk RNA sequencing, and they see that the MEK inhibitor significantly reduces their ETS2 module, all the genes that they found in the knockout and overexpression assays. Additionally, they have an inflammation score from a gene set, and they see that the MEK inhibitor significantly reduces that as well, and does so to nearly the same level as infliximab. So to summarize the paper, the authors identified this intergenic haplotype on chromosome 21, Q22, that is linked to 4 different inflammatory diseases: IBD, ankylosing spondylitis, Takayasu's arteritis, and primary sclerosing cholangitis. They identified an enhancer in this region that physically interacts with and promotes the expression of the transcription factor ETS2 in macrophages. Further, they found the pioneer transcription factor PU1 binds to this enhancer and promotes chromatin accessibility to facilitate interaction with ETS2. And having the causal variant that's associated with these diseases increases the activity of the enhancer, PU1 binding, chromatin accessibility, and ETS2 expression. They found that this enhancer and ETS2 are necessary for macrophage immune activity. It's a bit controversial what ETS2 is doing, and I think a lot of the prior studies were using macrophage cell lines like THP macrophages, and so this is really good, rigorous data showing that ETS2 is necessary for macrophage immune activity. And further, they show that genes regulated by ETS2 are expressed in several inflammatory and infectious diseases, especially diseases related to the chromosome 21q22 risk haplotype. And they identify MEK inhibitors as a potential therapeutic for inhibiting macrophage immune activity. I noticed also in a press release, the PI says that the group has made an antibody drug conjugate for targeting macrophage MEK specifically. So I'm very excited to see what comes of this. I have some comments, questions, and critiques, as always, with every journal club. The authors used many clever approaches, like in vitro transcribed mRNA transfection to overexpress ETS2. I thought that was very clever. Using the links database to find the MEK inhibitor. I really like that sort of approach. The authors leveraged publicly available data sets and databases throughout the report, which, again, I'm a big fan of. Some reviewers I know will poo-poo the use of these publicly available data sets, but I think it is really great when authors can discover new biology using published work, because then they don't have to use taxpayer dollars to do the experiment and, frankly, waste money. So I think we should be applauding people when they are using publicly available data to make new discoveries rather than critiquing them. A huge strength of this article was the use of primary human macrophages in all of their experiments. This is definitely more relevant than the use of THP1 macrophage cell line, which has been used in many studies. Another important thing about this paper was the effect size that they found with the MEK inhibitor in vitro was very good and equal to the standard of care. Now, I would love to see how it works in a preclinical model, but I understand the limitations of mouse models, DSS colitis not exactly reflecting IBD, but I would like to see how this works in a preclinical model before we put it into patients. And in terms of mechanism, it's still very unclear to me how MEK interacts with and inhibits ETS2. We don't necessarily need to know that before we move forward with this as an approach, but I think we need to understand how it interacts with ETS2 eventually so that we can develop more targeted therapies. And I would also like to see how it works in conjunction with the standard of care. Is it additive or synergistic? And again, I mentioned this at the very beginning. The article is really focused on these 4 inflammatory conditions. And even if you're not interested in those specific conditions, I think this paper is really important because this approach that the authors use, this combined genetic immunology approach, could be used for any disease, not just immunological diseases either, but any disease with a genetic component. So I think everyone should be paying attention to this, specifically the use of the GWAS data, because there is so much GWAS data that is not being leveraged. And that sums it up. Overall, really great paper. I learned a ton. I hope you all did too. So thanks for joining me today. Until next time.